friends and viewers, welcome to my session today. Myself, Dr.K. Narayanasamy, Associate Professor and Head, Department of Forensic Science, Nehru Arts and Science College, Coimbatore. So today in this particular session, I am going to talk about a very useful topic and an area where a lot of research is going on. It's nothing but genomics and proteomics. That is, the first few decades we are talking about the gene and genomics, and this genomics is always connected with proteomics. So a gene which is going to carry the information from your previous generations. For example, you see that you have a character that is there in your gene. That character is that transferred from your immediate parents or your grandparents. So genomics is going to talk about that kind of functionality of the gene. Then we will come to the other area, the other hand of your body, that is proteomics. If you have genomics one side, proteomics the other side. And proteomics is going to talk about the functionality of your gene. That is, a gene is expressed in terms of protein. That is called proteomics. So in this video session, I am going to talk about the genomics and proteomics. So we are going to see about genome maps and the different types of genome maps here. So in the background you can see a picture. It is the three important life critical molecules present in the living systems. One is DNA, the other one is RNA, third one is protein. All the three are important in central dogma. So what is this central dogma of molecular biology? It is having the combination of all the three molecules, DNA, RNA and protein. DNA can be duplicated into another copy. It is called DNA replication. So DNA can replicate. Then the information present in the DNA can be taken to RNA, and it is called transcription, where information coded in the sequence of base pairs in the DNA molecule is passed to molecules of RNA. Then the information which is there in RNA can be converted into protein molecule. This is called a translation, where information present in the RNA is passed on to proteins. It never passes from proteins to nucleic acids. That is, you see that protein to nucleic acids coming back is not possible. Only forward reaction from RNA to protein is possible.
Imagine we have a beautiful article with us. What if we want to make a copy of it? The best way would be to make use of a cast or a mold. Similar strategies are used by our cells. The most important task for a cell is to synthesize proteins based on the codes in DNA. But how are these codes read? What's the process that helps decode this language? And are proteins synthesized merely by the sequence of A, T, G and C? The answer to all questions lies in the central dogma of life. But what exactly is the central dogma? Well, DNA makes copies of itself by the process of replication. Post-replication in every cell, the code of the DNA is cracked and transcribed into a messenger RNA. This mRNA is then read and the code is translated into proteins. This interesting concept is nothing but the central dogma.
So all these three critical molecules, DNA, RNA and protein, are involved in central dogma of any form of living system. We are going to see about genome maps with this central dogma background. So what is this genome map or genetic map? It is a map that shows the relative location of two genetic traits, related or closely related traits. The way to do this is to use the offspring means F1, F2 generations of an organism and track how many times two given genetic traits are inherited together. They are inherited from one generation to another generation. For example, hair color and eye color, both are inherited. There are two general types of genome mapping. One is called genetic mapping, the other one is physical mapping. Both types of genome mapping, actually these two mappings are going to help or guide the scientist towards the location of a particular gene or the section of a DNA called a gene on a chromosome. However, they rely on very different information. And if you go for positional cloning and gene mapping, we are having a lot of strategies. So we have general mapping strategies. One is called genetic mapping, other one is called physical mapping. Then we have RFLP, restriction fragment length polymorphism linkage analysis. In that we have RFLPs linked to disease genes, then predictive value of linked RFLPs. Then gene isolation with linked RFLPs, where we have chromosome walking, then criteria for identifying a gene. Then physical mapping. This is physical mapping of a particular gene, where we have cytogenetic analysis, fluorescence in situ hybridization technique is there, FISH, FISH is there. So we have general mapping strategies like genetic mapping. What is the definition of genetic mapping? By definition, the ordering of the genes on a particular chromosome or chromosomes according to recombination frequency. So here genetic mapping may sometimes be required first before probes can be found to use in physical mapping. Then physical mapping by definition says that determination of physical distances between the genes, adjacent genes in base pairs of DNA, that is the distance, using cytogenetic analysis and molecular techniques. Physical mapping is used to ultimately pinpoint or exactly locate the gene of interest. So if you take a human genome, mapping the human genome can be done by all the three: genetic mapping, physical mapping and DNA sequencing method. So we have classical genetic linkage studies. So genetic linkage mapping, it involves determining the statistical association of specific traits with genetic markers on chromosomes using pedigrees and crosses. So for that we have several methods used. So we use recombination frequencies to determine relative distances between marker genes on a chromosome. Then genome-wide association studies called GWAS is there. Then humans require 24 different maps, one for each of the 22 autosomes and one each for X and Y chromosomes. Then marker alleles are used to determine the rate of recombination. Example, crossing over is there between the linked genes. So linked means on the same chromosome or on the same genome. So the unit which is used or measured for each linkage map is recombination frequency. That is nothing but recombinants divided by total progeny. This is reported as map units, MU, or centimorgans, CM. Different types of markers, they are used in genetic mapping. So genes can be used as genetic markers, but they are not ideal choices because they occur frequently. Example, every 100 kilo base pairs in human genome we have a marker. So greater marker density, it is usually required for identifying the genes. So three major types of markers are used here. One is RFLP, restriction fragment polymorphisms. This is actually substitutions at a restriction site, where in the restriction site restriction endonucleases are going to cut the DNA molecule. Then STR, very short or short tandem repeats STR, otherwise called microsatellites. Then SNPs, single nucleotide polymorphisms. Then STS, it is sequence staggered sites. So genome-wide mapping can be done. Here you see that high density genetic mapping, it was first revolutionized in the year 1980s by the discovery of abundant polymorphic genetic markers like macrosatellites and microsatellites. Then research teams, they collaborated and they added to a common database. Then in the year 1994 human genetic map, it had localized almost 5,264 microsatellites to 2,335 chromosome loci. So now it's the average density of one marker for every 599 kilobase pairs. So in this process, thousands of sequence tagged site, that is STS, were identified. STS, that is nothing but couple hundred base pairs of known sequence. Two types of physical mapping, physical maps useful for sequencing a genome. One is called low resolution maps. They are cytogenetic or FIS sketch maps. They are actually chromosomes can be stained with some dyes. So stained chromosomes, they produce banding patterns composed of bands that average 6 MB. Then regions are designated by their position relative to the centromere, which is there at the center of the chromosome. Q is equal to long arm and the P, which indicates short arm, numbered from the centromere starting with the number 1. So genes and other sequences are localized to chromosome maps with probes, and by using a technique called fluorescence in situ hybridization FIS fish. Various types of radioactive probes, radioactive isotope probes and stains also can be used to mark specific regions of chromosomes. Then it provides a physical map of the overall structure of each chromosome or the region of the chromosome. Two types of physical maps very useful for sequencing a genome. The second one is called a high resolution map, YAC or BAC clone contig maps. So mechanically shear or partially digest genomic DNA with restriction enzymes and clone large 200 to 500 kilobase where see that kilobases overlapping fragments to YORCs or BACs. So an entire genome or single chromosome can be represented in a YORC or by BAC clone library. It depends on the starting point where the gene starts. Then overlapping YAC or BAC clones can be assembled into a scaffold without sequencing by DNA fingerprinting using markers like microsatellites. So BAC vectors with a capacity of 300 kb and ability to replicate in a bacterium called E. coli, Escherichia coli, that have become popular for genome sequencing. Now we see that routinely sequenced using the shotgun method. Sequencing the human genome, here we have two major players. One is called SGP, other one is called CRA. What is SGP? Human Genome Project, publicly funded international consortium like NIH, DOE, etc. Then Francis Collins National Human Genome Research Institute, NHGRI. Then began in US in 1990. They started with a goal of 15 years. Then genetic and physical mapping approach and dideoxy sequencing was done.
The Human Genome Project was one of the great feats of exploration in history. The HGP was an inward voyage of discovery led by an international team of researchers looking to sequence and map all of the genes together known as genome of our species, Homo sapiens. Beginning on October 1st, 1990 and completed in April 2003, the HGP gave us the ability for the first time to read nature's complete genetic blueprint for building a human being. The HGP has revealed that there are probably more than 20,500 human genes. This ultimate product of the HGP has given the world a resource of detailed information about the structure, organization, and function of the complete set of human genes. This information can be thought of as the basic set of inheritable instructions for the development and function of a human being.
Then Celera Genomics Corporation, CRA, they did it. they had a spin-off of Applied Biosystems API. Then Craig went our CEO. Then they created this in the year 1998 with a goal of 3 years, direct shotgun method and deoxy sequencing along with HGP's maps for validation is done. Both groups collected blood and serum and sperm samples from anonymous male and female donors of different ethnic backgrounds. So for identifying genes in the DNA sequences, we have to do first step, that is called annotation step. That is nothing but identification and description of putative genes and other important sequences. The second one is called ORFs, open reading frames. So this ORF is nothing but potential protein coding sequence that begins with a start codon and ends with a stop codon. So all ORFs come in all sizes. So not all ORFs they encode proteins. 6 to 7% they will not code for protein that is done in yeast. Then ORFs with introns, that is called intervening sequences, can require sophisticated computer algorithms to detect it. Then sequencing the human genome. Two major players. We have the HGP and CRE, as I mentioned earlier. And identifying the genes, we are having annotation step first, then ORF second.
Friends, let us go for a small quiz now. You try to give answer. What is a genome map? A physical map showing the geographic locations of species, or a graphical representation of the arrangement of genes and markers on a chromosome, or a map used to navigate gene sequencing laboratories, or a database of genetic information from various species. Give your answer now. The answer is a graphical representation of the arrangement of genes and markers on a chromosome. Which type of genome map provides the highest resolution? Genetic linkage map, or cytogenetic map, or physical map, or metabolic map? Give your answer now. Which of the following methods is commonly used to create physical maps of genomes? Polymerase chain reaction (PCR), or restriction fragment length polymorphism, that is RFLP, or fluorescence in situ hybridization (FISH), or DNA sequencing? Give your answer. What is the purpose of a genetic linkage map? To display the physical distance between genes, or to show the location of genes on chromosomes based on recombination frequency, or to sequence the entire genome, or to identify mutations in DNA sequences? Give your answer. Friends, these two questions are true or false questions. Give the answer. Cytogenetic maps can be used to identify chromosomal abnormalities such as deletions, duplications, and translocations. Your answer is next one. Restriction fragment length polymorphism, that is RFLP analysis, was an early technique used in the development of genetic linkage maps. True or false?
Friends, I hope you have understood what is gene, what is genomics, what is a protein, what is the role of proteomics. So after this useful session, I hope that you may able to visualize or think about the protein structure, then the genomic function, everything. So with that, I extend my thanks to you all who have watched this video. So thank you very much.